Hello everyone, this is the Chinese Mitten Crabs aquaponics project. Welcome to our video about basic genetics. Today we're going to try to explain why you look like your parents. It's all about heredity. So what is heredity? Heredity is the passing on of characteristics from parents to offspring. Although family members are similar in many ways, there are still differences in some characteristics, such as height. The differences among individuals of a species are called variations, and the study of heredity and variations is called genetics. With the advancement of technology, scientists have discovered that inherited characteristics are determined by genes. But what are genes? Before answering this question, we need to understand the structure of DNA, which is short for deoxyribonucleic acid. Genetic information is carried in DNA stored inside the nucleus, and each DNA molecule wraps around some proteins to form a chromosome. As we can see, DNA is a very important molecule to carry out genetic information. Its structure is therefore crucial for carrying out its function. DNA belongs to a group of large molecules called nucleic acids. Nucleic acids also include RNA, which is short for ribonucleic acid. It is important in protein synthesis. So what makes up a nucleic acid? If you look at this diagram, you can see each nucleotide is formed by a phosphate group, a 5-carbon sugar, and a nitrogenous base. Nucleotides are joined together by hydrogen bonds between the sugar of one nucleotide and the phosphate group of the next, which results in a polynucleotide which bases branched out along the sugar-phosphate backbone. Although both DNA and RNA are included as nucleic acids, they have some differences in their composition. For example, DNA has deoxyribose as a type of sugar, while RNA has ribose as a type of sugar. As for types of bases, DNA has adenine, thymine, cytosine, and guanine, while RNA has adenine, uracil, cytosine, and guanine, which uracil replaces thymine in DNA. And finally, for the number of polynucleotide chains in a molecule, DNA is double-stranded, while RNA is single-stranded. Now, let us look at the Watson-Crick model of DNA. James Watson and Francis Crick proposed a 3D model of the DNA in 1953. They proposed that two strands of DNA are anti-parallel. The head of one strand is always laid against the tail of the other strand of DNA and twists around each other to form a double helix. Adenine of one chain pairs only with thymine of another chain by hydrogen bonds, while cytosine with guanine. These rules are called complementary base pairing. A gene is the basic unit of heredity. It's a segment of DNA molecule on a chromosome. The base sequence of DNA is a gene determines the amino acid sequence in a polypeptide in a way called the genetic code. Polypeptides are very useful, since they are responsible for forming proteins that perform various functions, including enzymes that catalyze reactions that happen in our body. Each inherited characteristic is controlled by genes. By determining the types of proteins a cell makes, genes are responsible for determining the body characteristics or traits of an organism. Let's look at another keyword, alleles. Alleles refer to a gene that can exist in two or more alternative forms. In a pair of homologous chromosomes, alleles occur in the same position with the position of the gene called gene locus. There are three features that enable DNA to be well suited to its function as a genetic material. First of all, DNA is formed by a large number of nucleotides, which allows it to carry a large amount of genetic information. Secondly, as many hydrogen bonds exist in its helical structure, DNA is a stable molecule. Last but not least, the pairing of specific bases enable accurate replication of DNA so that the same genetic information can be passed from generation to generation. Let's look at how a DNA molecule replicates. As the helical structure of DNA is held together by many weak hydrogen bonds between bases, the hydrogen bonds first break and the two strands separate, with each strand acting as a template to synthesize a new DNA strand. With the help of an enzyme called DNA polymerase, it catalyzes the addition of three nucleotides complementary to each template, with each having one original strand and one new strand. Two identical DNA molecules are formed. Since we have learned how DNA molecules are replicated, you might begin to wonder how characteristics of our parents get passed on to us. Here comes a genius, Gregor Mendel. He was wondering about the same thing too. According to Encyclopedia Britannica, Mendel was a botanist, teacher, and Augustinian prelate. He is the first person to lay mathematical foundation of the science of genetics, which we called Mendelism. He conducted breeding experiments on garden pea plants and observed their characteristics in successive generations. He proposed hypotheses to explain how characteristics are passed on from parents to offspring. As Mendel was interested in exploring monohybrid inheritance, he started off with experiments on the inheritance of one pair of contrasting characters, namely tall and short stems. This type of inheritance is called monohybrid inheritance because it involves a single characteristic controlled by a single gene. Mendel dusted the pollen grains of purebred tall pea plants onto the stigma of purebred short plants, and vice versa. In both cases, they found that all plants in the new generation were tall. The first generation produced from this kind of breeding is called the first filial generation, or F1 generation, where the word F stands for filial, which relates to the word family. Mendel then allowed the F1 generation tall plants to self-pollinate, collected and sowed their seeds. He found that offsprings in the second filial generation, F2, contained 787 tall plants and 277 short ones. The ratio of tall to short plants was about 3:1. The experimental work carried out by Mendel is an example of a monohybrid cross. Mendel also performed other monohybrid crosses in pea plants. Their results showed a similar pattern of inheritance, with only one of the two characters seen in the first filial generation, and the character which has disappeared in first filial generation reappeared in the second filial generation. The ratio of the two characters in the offspring was always about 3:1. Let's look at how Mendel interpreted his experimental results. Mendel proposed a hypothesis to explain the results of his breeding experiments. We're going to use the height of garden pea plants as an example to illustrate this hypothesis. The height of plants is controlled by a pair of alleles. We use capital T to represent the allele for tallness and lowercase t for shortness. The tall parent possesses 2 tall alleles, while the short parent possesses 2 short alleles. Their genotype are represented by 2 capital letters T and 2 small letter t's, respectively. During gamete formation, which is known as mitotic cell division, alleles separate so that each gamete receives only 1 allele from each pair. This is known as the Mendel's first law of inheritance, or the law of segregation. If you look at the diagram, you will see that the fertilization of gametes from the 2 parents results in the first filial generation with the allele combination of a big letter T and a small letter t. Individuals of the first filial generation possesses 2 different alleles for the height of plants. They are said to be heterozygous for this characteristic, and they are called heterozygotes. They are also called hybrid, as they are produced by crossing 2 homozygous parents, which are genetically different for the height of the plant. The phenotype, which refers to the observable character of the individuals in the first filial generation, is tall, because allele capital T masks the expression of allele small letter t. Allele uppercase T is said to be dominant, and allele small letter t is said to be recessive. The first filial generation can produce 2 types of gametes that contain either allele big letter T or allele small letter t. Therefore, there are 4 possible combinations of gametes when random fertilization of gametes from 2 first filial parents occur. This produces tall and short plants in the second filial generation in a ratio of 3:1. Let's look at other examples of monohybrid inheritance. Besides garden pea plants, many other organisms have characteristics controlled by a single pair of alleles. For example, color of grains in a cob of corn, length of wings and body color of fruit fly, and coat color of mouse. Not only so, normal ears versus short ears in mice are also one of the most common examples when we are looking at monohybrid inheritance. Humans also exhibit a number of examples of monohybrid inheritance too, such as the shape of hairline, earlobes, dimples, shape of little fingers, and thumbs. Fun fact, did you know that a curved thumb is called a hitchhiker's thumb? The graphical representation of a genetic cross is often useful when solving genetic problems. There are 2 common methods, namely genetic diagrams and Punnett squares. They are used to show how gametes from the parents may combine to produce offspring. This is the end of the video. Thank you for watching. Please give us a like and subscribe to our channel. Click on the tab to watch the next part of the video series.